Hello, everyone. Sorry, I came in late. Without wasting time, we'll start immediately. Last Monday, we looked at practical biology. What is involved? What do we expect? Having seen that it involves lots of drawing, we examine how to draw biological diagrams. We also looked at the views where we are drawing from, whether anterior, posterior, lateral, ventral. Then we examine the where we label drawings by the size using guidelines. Then we looked at a microscope, which is the major equipment we'll be using throughout the course of the exercise. Today, we proceed by examining lesson 2. At this point in time, that's procuring plant and animal materials for practical. So let me start by sharing my screen. Our lesson, once again, is procuring plants and animal materials for practical work. At the end, that's procuring plants and animal materials for practical work. Yes, sir. Yeah, cover no more. Let me just, this thing is not coming as supposed to. So let me start sharing my screen. Yeah, the first order again, the first. Oh, the first. No, that again, the first. Just there, the first. Oh, the what is happening? One point in the first. Sorry, my PowerPoint is have some problem. So at this point in time, I will start sharing my screen. So let me just start sharing my screen. Okay. The topic, once again, is procuring plants and animal materials for practical work. And on completion of this lesson, you'll be able to place the order with a dealer for purchase of specimen and live animals. Collect live animals from your natural environment. Learn how to identify male and female animals. Collect algae, bryophytes, pteridophytes, and gymnosperm. Collect, press, dry mount, label, store, and preserve angiosperm specimen to make a herbarium. Activity 1, you will look at how can you procure animal material. Give a thought to this. First, let's look at placing order for the specimen and live animals. Specimen are to be procured from the market. First, there are 2 letters to be written from your institution. The first letter is for inviting quotation from dealers. Then the second one is the letter of placing order. First, the section, procurement section of your institution will write a letter to dealers inviting quotations. When they must have sent in their quotations, they will now write a second letter of placing order, during which the lowest possible quotation, as the case may be chosen, but arrangements will be made in case of disappointment to switch over to another one. Then let's see collection of aquatic vertebrates. Required for such aquatic invertebrates, materials required: a net, fine woven for small animal and coarse woven for large animals. Then large clean jars or buckets. Then shallow white pans. See diagrams. For collection of insects, we use a sweep net, as the case may be, depending on the size. There are different sizes of the nets one can use. Then for collection of insects, we can use also flashlight torch or aspirator, as described here. When you flash your light on the insect, it will make them to immobilize, then you can pick them. You can use an aspirator. Then let's see collection of plants. First and foremost, for collecting a bryophyte, you first of all scrape the bryophytes from the place of occurrence with the help of sharp scalpel or knife, and keep them in polythene bags within which they remain alive to a number of days. Then keep these bags closely tied. Keep the bags loosely tied and in damp condition in a lab. Then wash the soil growing specimen with ordinary water to remove soil particles and dirt attached to plants. Then keep the bags under illumination at 0 to 5 degrees centigrade to keep the plants alive for a longer duration. Freeze dry the material, label and keep it in the cupboard. Then for pteridophytes, we collect the pteridophytes from natural habitats in mature spore producing states. Then collect the plants with or without strode belly or mature sporophyll in polythene bags, loosely tied at the mouth. Then if the material is large, cut them into pieces, fix label, and keep them in a cupboard. Then dealing with gymnosperms, we collect the root, the stem, the leaves, male and female gymnosperms of the plant. Then cut the material into small pieces of 3 cm, face, label, and keep them in a cupboard. You can collect the dry fruits or cone of gymnosperms and preserve them as such. Then looking at collection of higher plants for making a herbarium. In collection of plants, do not pick plant haphazardly. Choose only those plants whose flowers are large and whose organs are easily seen. If possible, collect complete plants with flowers or fruits for easy recognition of specimen of some families. The underground parts, root, bulb, rhizome, are often interesting. Try to collect them. If the plant is large, take just a branch with leaves, flowers, and fruits. Then carrying the specimen. Do not mix up or damage the selected plant with others. Place the plant quite flat between newspapers and place in a cupboard folder and fastened in a strip, or keep the plant vasculum. Then attach a label to each plant stating details of its dwelling place, dates, name of place, flower color, and any other interesting feature. Then examining the plants. Identify the name of the plant with the help of your course tutor. In due course of time, you'll be able to name the plant by yourself. You can also keep a plant with some labeling with it, and it could be identified with the help of the flowers. Then make a few sketches to point out the characteristics which help you to identify the plants. Then dry in between papers using dryer. Then in conclusion, in this lesson, you've learned that you either buy or collect animal material. Once materials for lab work have been acquired, they must either be killed and preserved for studying or kept alive in suitable conditions for the same purpose of being studied. You go to the natural habitats of the plants and find them. Most lower plants have to be faced before storing away in labeled sterilized bottles. For most higher plants, their preservation process is drying and the storage is in the herbarium. Next activity to be taken, describe how you will collect the following animal materials: 1 aquatic invertebrates and 2 insects. In next lesson, we'll be looking at preparation of temporary slides. Thank you for listening. I'll stop sharing at this point for us to reconvene. So we are back. Any questions or concerns?
No, ma.
No question. Is the lesson clear?
Yes, ma. I actually understand.
Sorry, it's like most people could not connect. Maybe they tried earlier. I was somehow late, but I had to deliver. I will upload it, even the recorded session, for them to watch in their own time. But I want to use this opportunity to announce that we'll be starting practical exercise on this course by next Tuesday, 3rd of October, 2023. And we have 13 designated centers for the practical in various political, geopolitical zone. North Central, we have Abuja and Makurdi. Northeast, we have Bauchi and Yola. Northwest, we have Kano and Sokoto. Southeast, we have Enugu, Owerri. South, we have Uyo and Benin. And Southwest, we have Lagos, Abeokuta, and Osogbo. I will encourage you. In fact, the faculty as a whole have put up notice of this practical exercise that will be starting next Tuesday. Please inform your classmates around, and you are expected to attend in the nearest study center to you. In the nearest study center to you is where you attend. Within the week, I'll put up discussion forums, and you are all expected to participate in the forum. While we'll be meeting again, let's make it 6:30 next Monday. Thank you very much, and see you next Monday. I will end at this junction.